afternoon. Thank you to Emily and the Foundation for inviting me to speak today and kind of really putting your trust in us to play a small part in this kind of therapeutic odyssey. So I'm going to tell you about drug repurposing for CTNNB1. I'll explain that in a minute. And we do use flies, and I'm going to choose to interpret your logo as fruit fly. I didn't get a chance to ask Emily what it means, but I'm going to choose. Well, I'm going to beg to differ. So, all right. So, I think many of you in the room know, parents know this, that rare disease medicine is precision medicine. With the state of science and funding, especially now, most rare disease therapeutic odysseys are pioneered by one or a few families, and even though this is a pretty large group, you were probably the pioneer family for your physician or for your pediatrician. And this article published last month in Nature Biotechnology highlights, I think, kind of the state of rare disease therapeutic discoveries right now. It emphasizes one of our screens and a bunch of other kind of discoveries, and kind of gives you a real good impression of where the field is right now. This was published about 2 years ago, and you can read about that here with some more QR codes. All right, so in my lab, we're really kind of pushing towards precision medicine drug screens in the lab. And what I mean by this is we want to be able to identify patients in the clinic, do a screen and find a drug for that patient and bring the drug right back to that same patient. So it's not a research project, it's not a 10-year process, it's how can we get to that drug as fast as possible. And to do that, we need fast models, efficient screens, and what we call drug repurposing. And before I kind of explain some of this, none of this is possible without kind of the close physician-patient partnerships we build with foundations like yourselves and many of the other foundations we work with. I think this is the only reason it works, and we do really the simple part, which is the science, and kind of executing and pushing forward is really the hard part. So first, fast models. Why fruit flies? Well, this time of year, you've got a banana sitting on your counter. You're swarming, probably right now while you're here, swarming with fruit flies, right? And they're a pest at home, but we love them in the lab. They are a premier biomedical model. They are fast. They have a short generation time, about 10 days. Lots of genetic tools. We've been using them for decades now in the lab. And they're complex like humans. They have a brain, a blood-brain barrier, a digestive tract, almost every organ system that humans have. And so that makes it a really, really exciting model. And for us, in particular, for these drug screens, we can do tissue-specific models of a particular disorder, and that really gives us the flexibility to generate an animal that still kind of displays the disorder in certain tissues but doesn't die from being too sick. Okay, but drug repurposing really is the star of the show here. And drug repurposing is really, really simple. It's finding an FDA-approved drug with an established use and finding a new use for it. Really, it's really that simple. And in our case, it's really just a new use in rare disease. And people think that FDA-approved drugs are really well-studied drugs, but in fact, once they're FDA-approved, people don't study them anymore. And in many cases, we actually don't fully understand what they're exactly doing in some of the disorders that they treat. You can think about lithium chloride for bipolar disorder, for example. We still don't really know what it's doing. So I like to think of FDA-approved drugs as kind of this very scary-looking iceberg, but it's a positive iceberg in this case. And above the water is what we think we know about these drugs, but below the water is really what we don't know, and really that potential. And we really want to tap the untapped potential for rare diseases like CTNNB1. So before I get into this, this is the incredible team of scientists that does the drug screens in my lab. They're full-time working on drug screens. We do anywhere between 4 to 6 drug screens at any one time with different foundations like yourselves. And they are very good at this. And I can't do any of this without them, so they do all the work. I just present it. So we started doing this about 4 years ago, and we've been building these bench-to-bedside rare disease partnerships with a number of different groups, with scientific consortia that are funded by NIH, with foundations like yourselves, and with our local Children's Hospital as well. And to date, we've done about 25 of these screens, and about a third of them are in some kind of clinical trial or N-of-1 type trial in the clinic, depending on what we find. And I think that it's having some really positive benefits for some of those communities. So I want to give you an idea of kind of how this partnership works and how we started with CTNNB1 and your foundation. So generally foundations contact us about wanting to do a drug screen. We've done a number of these screens now with a number of groups in Combined Brain, for example, and other groups. So we have a little bit of a name for ourselves. We have feasibility discussions. Not everything we can model on the fly, for example, and not everything makes sense to do a drug screen on. And then once we decide that this is something that we can do, we generate a model. The nice thing about flies is that there are a ton of off-the-shelf tools that we can use before we resort to generating new models. So we try to exhaust those options before we make new models, because it just gets more expensive at that point. We have a phenotype development pipeline where we screen about 30 different phenotypes, and we just agnostically pick the best one for the screen, and I'll tell you a little bit about that in a second. We have cost discussions. You know, those 5 scientists have to get paid, right? And so it costs money to do this. But once we kind of settle on all of that, we do the drug screen. The drug screen itself takes about 4 months. We did that just about 4 months for CTNNB1, and I'll kind of go through that again in a second. And once we finish, we start having discussions about the results, about kind of what the drugs look like, what they're doing, and what the possibilities there are. And with the top kind of candidate list, we run a bunch of validation assays, which I'll tell you a bit again later. And then all along the way, we're having clinical discussions about potential N-of-1 trials, or kind of testing in other models, or moving forward in a number of different ways to think about how these drugs might work. So initially, you know, we do a search for a phenotype, and a phenotype is just something that is wrong, that goes wrong when you eliminate the function of the gene. And what I should say kind of in the outset is that, you know, our screens are pretty different in that we don't care what we're trying to fix. We're not looking to increase beta-catenin per se, even though that could be the possibility. We're just trying to fix whatever goes wrong, right? If we can fix that, we know that it's hitting something that's important, at least in the model. And so we have a number of phenotypes that we like to screen on that we're really good at. For example, you can eliminate the function of the gene throughout the fly, and you can look at survival, for example. One of our favorite models and what we use for CTNNB1 is eyes. We can eliminate the function of beta-catenin, for example, in the eye, just the eye, right? And you can kind of screen on shape and size. We can eliminate you know, function in neurons or in glia, the support cells of the nervous system, and screen on behavior or survival, and we've done all these for different disorders. But for CTNNB1, we settled on screening on the eye, and beta-catenin is an incredibly well-studied gene, as you know, especially in flies. This whole pathway was kind of discovered in flies, and so this was really nice for us. But here on the left is a wild-type healthy eye. On the right is what happens when you eliminate CTNNB1 to just a little bit more than what we see in the patients. And that's what we call a rough eye phenotype. You can appreciate that it's really abnormal. It's very disorganized and much smaller than the control. And we actually use this quantitative size phenotype as our screening phenotype. And so here you can see we can just measure the size of the eye in pixels. And you can see in blue it's smaller than the control healthy eyes in red. And our screening is really simple. We give them drugs and ask, can we move these blue dots up towards the red dots? And so this is our workflow. We screen using the Presswood Chemical Library, which is a collection of about 1,500 FDA-approved drugs. That's roughly half of the known FDA-approved drugs. It covers all of the known classes of drugs, and represents kind of every kind of drug you can think of. So the flies are fed these drugs. They grow up on the drug, so they're on the drug their whole life. And we collect the eye data, we image it, and we calculate a score representing that phenotype. And so this is what the data looks like. This is 1,500 drugs on a single plot. So 4 months of work for 5 people. right? This is how it always is, right? So this is a z-score plot, and z-score is like a standard deviation, but for your purposes, everything above the red line is something that improves the phenotype, and everything below the red line on the bottom it makes the phenotype worse. So here we have 28 drugs that are known as suppressors, which improve the phenotype, and 48 drugs that are known as enhancers that worsen the phenotype. And this is really boring to look at, a plot of dots. So just to kind of show you what it looks like, in the middle here is an untreated eye, and on the top is an improved eye, and on the bottom is a worsened eye. And you can see that actually in the worsening side of things, it really wrecks the eye, some of those drugs. And so what do we hit? I'm going to say I'm not going to talk about drug names here, but as we work with the foundation, I'm sure you'll get to know this data quite a bit. So in the suppressor side of things, we hit a number of different drugs, actually. We hit drugs that hit the serotonin pathway, the histamine pathway, anti-malarial drugs, and drugs that target sodium channels. Some of these may be actually related to GSK3 beta. And on the enhancer side, what was really interesting was, usually this is a little bit more balanced. For us, this is actually the most unbalanced screen we've had, where there's quite a bit more enhancers than there are suppressors. But once you realize that what we were looking at is reduced beta-catenin, and there's a ton of drugs that are being developed to reduce beta-catenin activity for cancer, and that's what we hit, things that make it worse, right? And that's actually a really good proof of principle that this is a good model. So nearly a third of these drugs that make the phenotype worse are known drugs that reduce beta-catenin activity. So reducing it further, even worse, right? That includes non-steroidal anti-inflammatory drugs. We hit 7 of those. Those are known beta-catenin inhibitors. PPAR alpha activators, which are known inhibitors, as well as EGFR inhibitors, all known inhibitors of beta-catenin pathway. That's kind of an overview of the screen. Again, I'm not going to talk about drug names, but you'll learn about that soon. Another thing we really like to look at is structures of these drugs, and this is kind of just like a puzzle game where you look at the structures and ask, are there just common patterns that we see, and what does that tell us? For example, these are 2 hits from the screen, and without telling you what they are, I think you can see that they are actually very, very similar to each other, nearly identical. And we like to see this because this tells us that whatever the structure is doing, it's important across multiple molecules, and that's kind of reinforcing that finding. Here's another example where the top molecule is basically the left side of the bottom molecule, right? And if you squint, actually, you can see that all 4 of these molecules are actually quite similar. Again, telling us that there's something to whatever these molecules are doing in this particular model. Okay, so at this point, we finished this screen about a week and a half ago. So we're in the hit validation stage right now, and after this meeting, we're going to talk with the foundation to prioritize these hits. We can't validate all 28 of those positive hits, but we can prioritize them. We want to prioritize them based on mechanism of action. Maybe something inhibits GSK3 beta, or to that kind of flavor. We also talk about availability. Some drugs are much easier to get than others. For example, we have 3 drugs that are over the counter. One's a common allergy medicine, one's a common heartburn medicine, and one's a common antifungal medicine. Again, no names for now. But we've had experience with other screens where an over-the-counter drug, where you can just buy in the grocery store, is the drug we're trying in the clinic and is working really, really well. So that's one reason why we really like to push on these at first, if possible. We always order. We also want pediatric safety, right? Not every drug that's FDA approved has pediatric safety or has ever really been used on kids, and so we kind of want that in the mix as well. We always order drugs from a new source, a new company, to make sure that everything's as expected. We run multiple concentrations, known as dose curve, that tells us whether something is better at higher dose or sometimes actually better at lower dose. We'll first run it on all these eye phenotypes, and then we'll run it on other phenotypes. I told you at the beginning that we did a phenotypic search, so we actually have a number of different phenotypes that this model produces that we didn't use for the drug screen. For example, when you eliminate beta-catenin in the wing precursor models, you get this really nice notch wing phenotype on On the right here, you can see that it's missing a little part of the wing. That's a very obvious phenotype that we can look at whether drugs can correct a totally unrelated tissue phenotype situation. So that's where we're at, and we're really excited to try to use this kind of really humble model, I think, to try to bring some hope to your community, to the rare disease community, and thank you for putting your faith in us, in playing our small part. You can contact me on that QR code there. And on the bottom is a QR code to a documentary we participated in that talks about how Drosophila fruit flies are playing a role in rare disease research. So if you're interested, you can watch that. Thank you.